Okay, so this video we will now start looking at the MHC II receptors. But before we get into this, let me clarify just a little bit more on the previous video for the MHC I receptors. I don't think I was really as clear as I wanted to be on some of those. Let's back up. We have two basic pathways in which an MHC receptor can post something. The first receptor is going to be, or the first pathway is going to be called endogenous. The endogenous pathway. And what the endogenous pathway is doing, we're looking at posting self-antigens, or those amino acids, onto an MHC I receptor. It will also post the peptide formation of a virally infected cell. Okay. In either case, we are taking the antigens from inside of the cell, posting them on an MHC I receptor. So if you recall, we had our cell here, and it took, these are peptides from inside of the cell, and it ended up posting them on a receptor out here. Okay, so now it went through the ER, and then it goes to this direction. That's just very basic. But you can see this came from inside of the cell. Now, I think I limited at that. What we could also have done is we could have also had a viral issue. So let me just do red. Okay, so this would be a virus that has infected the cell. The virus would have then gone to the endoplasmic reticulum, that's ER, okay, which would then through the Golgi and then get posted on a platform as well. And since we used red for virus, we'd put red there, okay? Either case, these are MHC I receptors, okay? So those are self-antigens or virally infected cells. The exogenous pathway is where we're taking something from the outside of the cell and bringing it into the cell. So you're going to be looking at like a macrophage or a dendritic cell, something that will phagocytize something, and then that brings it into the cell. Okay, so well, let me just stop there. So it's bringing something in from the outside, pulling it into the cell, and it will post it on what's now an MHC II receptor. So let's look at what that one is doing. So if I can scroll around here. Okay, so let's look at what that's doing. So I've got my standard cell on this one, okay? And let me use blue as, say, bacteria. Okay, so we're going to have a bacterium on the outside of the cell. And let's say this is a macrophage. Okay, so a macrophage is going to go around and chomp on things that it can't find. Cellular debris, bacteria, whatever the case is, it's going to eat this up. So let me kind of just draw something here. And I guess we can draw it. We can kind of come this way. Not quietly attach it yet. And let me erase a little bit so you can see that the cell, in this case, is going to try to phagocytize this component because these two parts will end up joining. Oh, that's still on the eraser. So these will end up joining. Let's go back to the eraser. And as you can see, this is going to be brought into the cell. So we're going to have basically this structure with the bacteria in it. Okay? That's your basic phagocytosis. This structure here is called a phagosome. Okay? So let me back that up. So this is called a phagosome. Okay, that's what that structure is. Let's go through the process of what's happening at this point. So remember, we have our, let me break it down this way. So let's say we have our endoplasmic reticulum here. So this is your ER, rough ER. Now, what does the rough ER synthesize? What does it make? It makes MHC platforms. Well, we spoke on the last video that it makes MHC I platforms. Now we're also making MHC II platforms. Both the same. Well, two different platforms, but the rough ER makes them both. Okay? So I will do, since we did, let's just do green. Let me change colors. Let me do pink. Okay, so we have, let me just do a pink platform on this one. Remember on the other one we had the green. If you can see on here, we only had that one stalk on here. It's not what they look like, but just to help delineate between MHC I and II, I just have these in green, and then I have one branch on this one. So you can see down here, it's pink, and then I've got two branches representing MHC II. This will be more relevant when we get into a few slides or a few videos from here. Okay, so the rough ER manufactures the MHC platforms, in this case MHC II. So let's do a couple different things. Let's take this one to the next step, and we break down that bacteria. We tear it apart. Okay? So these are all the peptide formations that need to be used. What happens at this point is this MHC, let me go back to black on this one. Okay, so the I wanted black. Black. Okay, so this is going to be in, and it goes to the Golgi. Okay, just like the other one. And then the Golgi buds off where it's going to, let's see, let me, goes to one of these. We're getting smaller and smaller on these pictures. Didn't mean to do that. Sorry about that. So now we have a small piece of a peptide on this one. So rough ER sends the MHC II platform to the Golgi. Golgi then merges with the destroyed peptides from the bacteria. It presents that, and then what we do out here is now we present that one and we had blue, okay? And it presents it this one. So this is an MHC II platform. Now remember, we had an MHC I. What color did we have before? Okay, so we had orange. We could also do Okay, so these were MHC1 platforms. And so we had this at least color coordinated. These are self antigens. And this one was viral. But both were MHC1. And then this is MHC II. So MHC II follows phagocytizing something, bringing it into the cell, tearing it apart, merging it with an MHC II platform, and then posting it on the plasma membrane. At this point, what happens is you are going to have T cells, B cells, helper T, okay, that all three of these, they're basically coming around, checking those out. They're checking those things out to see, hey, do you belong in here or not? If you belong in here, you're good to go. If you don't belong in here, we're going to destroy you. So that's the basic idea of this. The next video, what we're going to look at is we're going to look more specifically at the B cells, the T cells, and where they're produced, where are they taught to recognize self. So we're going to go through that whole process. If you don't understand this this portion right now or the previous video, please go back, review that, understand this portion, because everything else from here builds on this. So if you don't understand this, the other stuff's not going to stick as well. So here we go. Let's get started.